Hi everybody, this is our genetic mutations mini lesson. Yesterday we explored the topic of protein production. You guys learned how proteins are made, what functions they serve, just how important they are. You learned about the processes of transcription and translation, and you even had a chance to practice some of this. So today we're going to talk about what happens when this process does not really go as planned. As this video is playing in class today, I do want you to have your document called Mutations open, and you should be going ahead and filling this in as we go. This video is embedded in the document as well, so if you want to refer back to it, you will have that available to you. All right, so understanding genetic mutations, changes in our DNA. What are genetic mutations? So these are changes to our DNA sequence. They can be small, like one base pair, or they can be many, many, many base pairs. These are kind of like typos or misspelled words in our DNA, which are the instructions. We can have them happen randomly, or they can be caused by environmental factors, or they can be passed down from your biological parents, so they can be passed down genetically as well. Over here in the image, you can see that I have a picture of our base pairs. Our A always pairs with T, G always pairs with C, and there are 4 base pairs: thymine, adenine, cytosine, and guanine. All right, so types of mutations. We are going to take a look at 5 different types of mutations. We have insertion mutations, which are when we have an extra base that's added in a DNA strand. We can also have this happen in chromosomes. There can be an extra chromosome or part of a chromosome. That can also happen. We've got deletion, so we've got the base is removed from the DNA strand. We can also have this in chromosomes. Then we have a base substitution. That's when one base is replaced with another. Duplication. We have a section of the DNA, a section of the gene or the protein that are copied or repeated, and we can also have this happen in chromosomes too. And then lastly, we have what are called inversions, and these are when certain genes or bases are switched or reversed. And we're going to take a look at some examples of these in just a moment. But what's important to remember from yesterday's lesson and then carrying over to today is the question is, what happens to the mutation if a mutation changes the structure of a protein? And the answer is very simply that it changes its function, because remember from yesterday, structure determines the function. If a protein is changed in such a way that its structure has been altered, it cannot do the job that it was meant to do, and it may do another job or it may just be useless. All right, so we're going to take a look at the same genetic sequence a couple times. So you're going to see this same pattern, CTAG. We're going to use Miss A's beautiful drawings again because they are very helpful, and I'm thankful that she has created them. So here we go. This is our normal sequence that we're going to look at. And remember, this is a very small hypothetical example of these. In real life, they would be likely much bigger than these. But for our example for our class today, we're going to be using some smaller ones. So an insertion. So here we have the insertion. Extra base pairs are added to the sequence. This can also happen in chromosomes. So here you see we have an extra G is added into our sequence. Then we have our deletion. So same initial code. We've got one or more bases that are removed, and then this can also happen in chromosomes. In this case, we've got C, T, A, and then our G was removed. So that is an example of deletion. And here we go with base substitution. So in a base substitution, we're going to have one base is replaced by a different base. So in this case, we have the G is being replaced by another A. Next, we have our duplication. So the section of the gene or protein that are copied and repeated. Remember, this can also happen in chromosomes. We can have a duplicate of a whole chromosome. In this case, we've got our CT are repeated. So it goes CT, CT, and then AG. And lastly, we've got what's called an inversion. So this is when certain genes or bases are reversed. So in this case, our T and our A are reversed. So those are some visual examples of all the kinds of mutations that we are going to be exploring. So let's talk about some causes of these mutations. Why do these things happen? So there's lots of different causes. One of the main causes, especially for humans is UV radiation from sunlight. That is one of the main number one causes of skin cancer and can be pretty detrimental to our health. Things like chemicals in your environment, copying mistakes made during cell division. You can have radiation exposure. Viruses, some viruses can be the cause of mutations, and then other times it's just random. So we have lots of different causes of gene mutations, but all of them are significant. And then what can the effects be? So we've spoken about this a little bit before, but let's just talk about it one more time. They can be harmful, meaning they can cause diseases or some kind of disorder or some kind of alteration that is not desirable. They can be helpful. Sometimes they can be advantageous. In the case of when we spoke about color differences or color blindness, some people might think it's an advantage to be able to see colors that other people can't see. Some can have no effect at all. In fact, most mutations are in fact neutral. They can affect protein production, and then we can also have them influence traits and characteristics that are passed down. And then some other real-world examples, a few of these we've talked about, some of them we haven't, would be something like sickle cell disease. That's a genetic condition passed down where the red blood cells are misshapen, and they actually do quite a bit of damage as they go through the body and through your veins. They do damage to other red blood cells and make them not do the job they're supposed to do. Color blindness, like we spoke about in a couple of our videos before. Something like lactose intolerance can be caused by genetic mutations. We can have antibiotic resistance in bacteria. And then even something like different eye colors can be caused by a genetic mutation. So the last thing I want to talk about is, why do we care about all this? Why does it matter? And it matters a lot, and these are some of the more important reasons why. So it can help us understand diseases. We can use it for medical research. Remember what we talked about. If we can find the cause of a disease or a disorder, scientists can possibly stop it before it even starts, thereby, eradicating it. We can use it to develop new medicines. We can explain the differences between certain organisms, and then also genetic mutations are responsible for a lot of the diversity that we see in our human race. So they're pretty important and a very interesting topic. So we were going to explore this a little bit more, but this is the mini lesson for today. Again, remember, if you have any questions, you can email me and I will be happy to help.